easiest way to study abroad. Guys, you don't need to worry about finding information on studying abroad. AECC will provide all the information you need. AECC has been helping students from all around the world to realize their dreams of studying abroad and has industry partnerships and local know-how to help you get set up with things like banking, insurance, accommodation, and more. The easiest way to study abroad. Guys, you don't need to worry about finding information on studying abroad. AECC will provide all the information you need. AECC has been helping students from all around the world to realize their dreams of studying abroad and has industry partnerships and local know-how to help you get set up with things like banking, insurance, accommodation, and more.
Hi everyone, thank you for attending this session today. So I'm Doni from AECC Indonesia, the moderator of today's webinar. So today's webinar will talk about the microscopy and imaging application for health and biopharma, delivered by Professor Jeremy Simpson from college principal and Dean of Science from University of College Dublin. Without further ado, please welcome Professor Simpson.
Thanks very much, Donny. Hopefully everybody can hear me okay. I'm just going to share my screen now and we'll get the webinar started. Okay, so very good afternoon to you all. And the webinar that I'm going to present today is relating to biology and health sciences, and it's entitled Microscopy and Imaging and their applications for health and biopharma. And what I would like to do in the session today is, I suppose in a way, give everybody who's listening to the webinar some insight into what the current health and biopharma sector looks like. And specifically, if you were to do a postgraduate degree in the area of life sciences, what might be the applications ultimately that you'd be using this information to do? So my name is Jeremy Simpson. I am the principal and the dean of the College of Science in University College Dublin in Ireland. But actually, for the purpose of today, I'm a biomedical science researcher. What I'm going to do is take you on a story as to how we use imaging and advanced microscopy in our laboratory every day to try and solve some important questions in health and disease. And really, I suppose the grand challenge that we are interested in, many other researchers are interested in around the world, is the grand challenge of drug delivery. We all know that there's some great drugs, some great pharmaceuticals out there, but the question is, how do we ensure that those pharmaceuticals are delivered to the right place in the body so they can have the maximum impact and really improve our health? And of course, one of the things that we know is that when we take a drug or a pharmaceutical, it's passed into our circulatory system, and that drug is passed all around the body. But actually, there's a problem there. And the problem is, of course, that drug doesn't really know where to go. If, for example, we're trying to target a specific, a highly localized cancer, how does that drug know which are the cancer cells? How does it know if it reaches those cancer cells, where to go inside those cells, and ultimately then kill those cells? And really, that is the big question that my lab and many other labs around the world are trying to solve. And we're doing this, or we're tackling this problem through the use of nanotechnology, using nanoparticles, tiny nano-sized objects, as specific delivery vehicles to carry drugs to very specific locations in cells. And I'm going to tell you a little bit about that story as we go through the webinar today. Now, what is common across everything that we do in my lab and indeed in many labs around the world is we thinking about how cells are organized in the context of tissues and organs within the human body. And what you can see in the slide here on the right-hand side is a picture of a small piece of human tissue. And it's only maybe 1 millimeter by 1 millimeter in size, but every one of those small green dots is an individual cell in that piece of tissue. So straight away, what we see in an image like this is that even in a very tiny piece of tissue, already there's a very complicated arrangement of cells, many different cell types, all jostling for space within that environment. And again, what I would like you to imagine is, for example, we are trying to deliver a drug to a specific set of cells within this piece of tissue. So if we now look at that on the left-hand side in the schematic, what you start to see is that there are many different types of cells. There's epithelial cells, for example, at the top that act as barriers to keep drugs out of the tissue. And then there's other different types of cells below, including cells of the immune system and cells of associated with blood capillaries and so forth. So, of course, one of the problems that we have when dealing with something like this is that tissues like this are very hard to work with. They can't be easily manipulated. And as you can see here, it's really not very easy to see the internal structures of individual cells. So clearly, we need to be looking a little bit deeper. So as a cell biology lab, we're interested in the individual cell, and we're interested in how the different compartments within individual cells, within tissues and organs are organized. So very simply, very schematically, we can look at a cell that looks something like this. And straight away, we can see that inside that cell are many different compartments. And for example, what we also know is that these different compartments inside cells are all highly connected. So for example, now with the yellow arrows, you see the pathways by which cells are able to move material from the inside of the cell to the outside of the cell. At the same time, now as shown by these green arrows, we can see some of the pathways that are able to bring material from the outside of the cell to the inside of the cell. And similarly, there are other pathways in blue that provide interconnections between all of these different compartments. And what my lab has been doing over many, many years now is trying to understand how these different pathways in individual cells work. And we think this is the key to unlocking our understanding of certain human diseases, and also importantly, how drugs and therapeutics and pharmaceuticals can be really precisely delivered into the cells. So of course, in order to be able to do this, we need to be able to visualize cells. We need to be able to see them in the lab and see these individual compartments. And so in the webinar today, I'm going to talk about some of the imaging approaches, the microscopy approaches that we've been using in the lab to try and solve these problems. So firstly, we're going to talk a little bit about some of the key techniques that we would use in my lab. And the first example is just simple time-lapse imaging. And hopefully you can all see in the webinar in the upper left-hand corner, this is a time-lapse movie taken over around 3 days of cancer cells just dividing in the lab. So this for us is a really nice tool at the high level, just to be able to see what would happen to a series of cells if we, for example, add a drug or a pharmaceutical to them. We can see if they continue to divide like this, or whether the addition of a drug may stop them dividing. So visualizing whole cells or population of cells like this is one really important tool that we can potentially use. What else could we do? Well, of course, one of the problems with this movie on the top left is we can't see the individual compartments within the cell, some of those compartments that I showed you in the previous slide. So therefore, we also need tools to be able to do that. And the tools that we use in my lab are fluorescent tools. We take the proteins and other molecules that we're interested in, and we attach fluorescent molecules, such as, for example, the green fluorescent protein or the red fluorescent protein, to the things that we want to study. And this now allows us to visualize individual proteins, even individual molecules in living cells. Now, when we bring these two techniques together, what that effectively means is that we've now got the ability to visualize specific molecules or specific proteins in individual cells. And that's now shown in the movie at the bottom. So here in this movie, it runs again, there's about 6 cells that we can see. And here we're looking at one specific compartment that we've labeled with these fluorescent tools. And then again, over the course of the time-lapse movie, we add a drug to these cells, and we see how these molecules change their distribution over time. So this method, this technology, now is giving us insight into what's going on inside individual cells growing in the lab. What else do we need? Well, in addition, of course, we're interested in the big picture. We're interested in all of the genes and all of the proteins, how they help organize cells, and importantly, how they help organize some of those pathways that I showed you earlier in the webinar. So what that means is we also need certain tools and robotics to help us carry out experiments in large scale. If we're going to do time-lapse imaging like I've shown you, we potentially need to do that on hundreds of thousands of cells, meaning that we have a large number of experiments that need to be established. So again, in my lab, the way we approach this is we use automation of a number of assays to really help us do experiments on a large scale. So of course, the output of this then is that we end up with many, many experiments. We take lots of images of the cells, but now, of course, we need sophisticated image analysis tools to make certain measurements within the cell. And that's what you see here on the upper right now. You're looking at an individual cell, and here we're using certain computer tools here to make certain measurements as to what's going on inside those cells. And then really the final piece of the puzzle are those cells themselves and the models that we use. And in this respect here, I've spent a lot of time recently developing more sophisticated cell models. And in this last example image that you see down here, this is a 3-dimensional assembly of cells. Effectively, it's copying what a small cancer tumor would look like in the body. But again, we're able to grow these in the laboratory environment, and we can then see how actually drugs would affect a structure like this. So that gives you a taste of some of the techniques that we use. But actually, what I also want to message you today is how the science that I'm going to tell you about is done by young people. It's done typically by researchers in their 20s and their 30s who are really great thinkers, who are really great innovators. And what I'm going to do over the next 5 minutes or so is introduce some of those amazing scientists to you. These are the scientists who work in my lab every day, trying to help me solve some of these questions in health and disease. And the first of these scientists I'm going to introduce to you is Adele. And Adele's work is specifically focusing on cancer, and specifically focusing on ovarian cancer. And what Adele is doing is she is actually taking cells from patients who have ovarian cancer. She's bringing those cells into the laboratory, and she's using these sophisticated imaging techniques that I've introduced to you to understand what happens to those patient cells when they're treated with certain anti-cancer drugs. And of course, this is a very powerful technique because it means that we're able, in principle, to study many, many of these mini tumors in parallel, to study each one of the cells in turn, and start to examine what's happening inside the cells with subcellular resolution. What else are we studying in the lab? Well, in my laboratory, we're also interested in how dysfunction of certain compartments in the cells contribute to human diseases. What you can see in this diagram over here is this is another map of all of those pathways that connect the different compartments in the cells. And specifically, what you can see here is a family of master regulators that really help organize these pathways. Now, these master regulators are called or termed the RAB-GTPase proteins, and they're really critically important passing material between the different compartments. But of course, invariably, what happens is some of these molecules go wrong. And when they go wrong, when they're mutated, for example, consequence can be human disease. And again, a number of the students in my lab, the PhD researchers, are working on these genes and their associated diseases. So for example, down here at the bottom, a gene or a protein called RAB33B that is involved in function of the Golgi apparatus. This is being worked on by Sophia. And what Sophia has found out is that when there are changes or mutations in this particular protein, it leads to a very debilitating skeletal disease called Smith-McCort dysplasia. And so Sophia's work is trying to understand how mutations in this one specific protein lead to this crippling disease here. And again, by studying cells using microscopy, as I've illustrated earlier, she's slowly working out how this protein actually works inside cells. A protein that's highly related to RAB33B is another one called RAB6. And this is being worked on by Lucy. And it turns out that this protein is another master regulator of the Golgi apparatus. And this protein plays a really important role in maintaining the general health of the cell. And in fact, all proteins that cells make that have to be passed to the cell surface have to pass through this compartment. And again, we've now established that there's a number of human diseases linked to this protein not working correctly. And then the final example on this page here of a specific protein that we're working on is RAB11. This protein is found out at the very edge of the cell. It's found in a series of membranes called endosomes or recycling endosomes. And this is work that's being carried out by Tierna in the lab. And again, what we know here is that when this protein doesn't work correctly, cells have the wrong, incorrect number of receptors at the cell surface, and this can lead to many different cancer types. So again, Tierna in her work has been trying to identify all of the interacting proteins of RAB11 to again understand how these proteins work in disease. So I think those are three very nice specific examples showing you how we're able to use imaging and microscopy to study specific diseases. But of course, what I also want you to take from this slide is that now here we have three different proteins from the same family that are all potentially causing disease. So sometimes it's also important to step back, look at the whole disease family together. And this work is being done by Unmani in the lab. And actually, Unmani is not really a biologist, she's a data scientist. And she is compiling data from our own lab, from many other labs around the world through various genomic resources to really use a data science and systems biology approach to try and make links and connections between all of these RAB family members to understand together how they potentially contribute to disease. So those, I think, are some really nice examples of studying specific diseases. But ultimately, of course, what our aim is, what our goal is to be able to treat diseases. And of course, to do that, we need to be able to know both how cells are organized, but also how to deliver drugs into cells more effectively. So we've been approaching this work by, again, developing a number of methods in the lab that allow us to do very high resolution imaging of cells in an automated way, and specifically use this technology to really understand how cells are organized. Again, we've been doing this in two ways. On the left-hand side, we've been doing this using conventional cell culture using monolayers. But more recently, we've been using what we call 3D cell culture using so-called spheroids to develop more complex models of how cells are organized. And again, we've developed technology that allows us to grow thousands and thousands of these mini tumors. This is what you can now see here in small arrays like this. Again, allowing us to systematically understand what's going on inside each cell of each one of these mini tumors. I have a number of students who are working on this technology. Both Maga and Danielle have been developing some of these new cell models over the last period of time and developing methods that allow us to understand how cells work in this very complicated three-dimensional environment. So ultimately, what does that mean? Well, it means that we now have effectively the technology, the principle within the lab, study how different drugs and different therapeutics carried using nanomaterials could potentially pass through the bloodstream and enter a cancer tumor. And so Alana and Maeva in the lab are specifically doing these experiments. They're growing mini tumors in the lab, and they're treating these mini tumors with nanoparticles containing a therapeutic allowing us to understand what's going on in this complex cellular environment. Of course, there's still one further tool that we need here to do these experiments. And this final tool that we need are the nanoparticles, these delivery devices themselves, the little red dots that you can see in the diagram here. And again, we're very lucky that we're able to image cells. So you can see image spheroids. You can see a hollow spheroid here. And what we're doing is we're then treating these mini spheroids with custom designed nanoparticles carrying drugs. And it's Hen's work in the lab as a synthetic chemist to develop novel functionalized carbon-based nanomaterials for drug delivery into these models. Then the final person who I'm going to introduce to you in the session today is one of my newest students, and her name is Madeline. Here she is here. Madeline is working on a new imaging technology called soft X-ray imaging, and her project is specifically using soft X-ray imaging that provides us now with much higher resolution, ultrastructural level detail of how nanoparticles enter cells. So what I hope you see from this first part of the webinar is how all of the students in this area, combining different overlapping methods and technologies, all centered around advanced imaging to help and to analyze critical problems in health and disease. So let's look at this in a little bit more detail. How do we actually go about doing these experiments every day in the lab? Well, of course, what I've described to you is that we're an imaging lab. We take images of cells, typically of cancer cells, and we use fluorescent tools, fluorescent molecules, to, for example, highlight certain things of interest. So in this example here, you can see these are two cancer cells. In blue, we see the cell nuclei. In green, we have one compartment labeled, red, a different compartment labeled. And for example, what we would do to these cells is we would add a drug or a therapeutic. We would then analyze these cells using very sophisticated imaging equipment and extract a number of features that describe what's going on inside these particular cancer cells. And we're able to do this with not just a few cells, not even just a few thousand cells, but actually potentially millions and millions of cells. We do this because we have a series of fully automated microscopes in the laboratory that are capable of running 24/7. So they are imaging around the clock, basically running our experiments for us. So in a way, this provides a very, very powerful platform for us to design experiments using cells, to take high quality images, and to extract really detailed features of the cells, hopefully to solve some of the problems in health and disease that I've talked a little bit about earlier. So let me give you some specific example. And the first example I'm going to give you is how we use this information that we gather from cells to improve how drugs and medicines, with the help of nanotechnology, can be delivered into cells. So of course, the first question is, well, how do we make these drugs smart? What are nanoparticles? What is nanotechnology? Well, nanoparticles can be considered nano-sized objects. So something between 1 and 100 nanometers in size. Just to put that into context, if we talked about, for example, a tennis ball, that would be 10 to the power 8 nanometers. If we talked about a salt grain, that would be 10 to the power 6 nanometers. In fact, the nanomaterials and the nanoparticles we work with are in the same size range as proteins, DNA molecules, and small viruses, between 1 and 100 nanometers in size. And we can build these nanoparticles in the lab out of lots of different materials, out of lipids, out of polymers, out of metals, for example. And this gives these different nanoparticles different properties. And importantly, what it allows us to do is to take these nanoparticles and functionalize them with different information. So for example, we can change the shape and the size of these particles. We can change the drug that they carry. And of course, importantly, we can change the material depending on the type of disease and the place within the body that we want to send these nanoparticles to. So we're very excited by this technology because we see it presents an opportunity to effectively make drugs smart, to take drugs that maybe we've been using for many years, but that are very toxic or very dangerous to our normal cells, to couple them with these nanomaterials and hopefully direct them to very, very specific places and thereby help deliver drugs to help cure certain diseases. But of course, this work comes with a big challenge because, of course, inside the body, our cells are arranged in very complicated patterns, as we looked at in those first couple of slides where I showed you the picture of a small piece of a human piece of tissue. And of course, again, the problem that we're trying to solve is how do you take a drug or a nanoparticle that is passing through our circulatory system, ensure that it's then delivered to, for example, a tumor cell in the right way? Well, of course, if you now think about everything that I've described so far, my lab has developed a series of technologies that in principle allow us to do that. We have, for example, automated microscopes that can visualize this process. And using these microscopes, we can grow these three-dimensional models of cells, and we can analyze all the different cells that exist in the different layers of these mini tumors. So in our lab, what we do is we grow these mini tumors in the lab. We start with simple single cancer cells, and under certain conditions, we can force these cells to aggregate and compact together to generate these mini tumors, or spheroids, which is what we often call them. And then we can add our fluorescent tools that we talked about to visualize things that are of particular interest. For example, the cell nuclei, the membranes of the cells, or indeed the nanoparticles that are entering those cells. And over the last few years, we've developed a whole range of technologies that allows us to grow these spheroids in a really high throughput manner. And the two methods that we use the most are the two that I've highlighted here on the top right, which is one of the main things that we do is we grow these spheroids in a scaffold or a matrix that then supports their growth. And again, this is very powerful because this would be similar to how the tumors would be growing inside our body. The second method that we use, as shown just below, is through the use of micro-patterning technology, where we create small micro-patterns on the bottom of plates, allowing us to grow the spheroids in arrays. And that really helps us, again, do the kinds of experiments that we want to do. So what do these experiments actually look like? Well, if we were to take a 96-well plate, here is a picture of it at the top, and we were able to zoom into one of these wells, you would see all of these little white structures inside the well. Each of these little white structures is a spheroid, is a mini tumor. So on average, we would grow about 700 of these mini tumors in a single well of a 96-well plate. And we now use our high-resolution microscopy to zoom in on each of these, and using our fluorescent tools, we're now able to visualize these. Now, that's what you can see on the right-hand side. So we can image these, again, from all different directions, and therefore now start to make measurements at the whole population level, make measurements from each individual cell within the spheroid, and start to make subcellular measurements so we can understand the trafficking pathways between those individual cells and within those individual cells. So what you can now see in this slide is, again, what data from these experiments look like. So in the top left, you see the raw data that the microscope gives us, for example, labeling the cell nuclei and labeling one of the organelles of interest, the lysosomes. And then what you see in the second, the third, and the fourth image are pictures of what the computer software sees. It's able to identify the cell nuclei, the cytoplasms of the cells, and ultimately each of the compartments of interest. And all of this is being carried out in 3 dimensions like this, making measurements for each individual cell in a volumetric manner within each of these mini tumors. So what are the things that we can measure in these experiments? Well, of course, the first thing we can measure is we can measure what happens to these tumors over time. We can measure their rates of growth. Remember, of course, they start from a single cell that divides into 2, 4, 8, 16, and so forth. So we can now nicely make measurements of how many cells are within these spheroids. And again, you could imagine that we could add certain drugs to these and see if this changes the cell count or the size of the spheroid. So we can measure the number of cells, but we can also measure the diameter of the spheroid. And again, this can be a very useful measure to tell us how quick the spheroid, how quick the tumor is growing. And finally, we can measure things such as the roundness or the sphericity of the tumor. And again, that can be an important measure to tell us when something is changing within the body of the tumor. Now, of course, what we want to do is we want to add our drugs, our therapeutics that are coupled to nanoparticles, they're shown here in red, to these particular tumors, to these particular spheroids. So you can see in red here, these are the drugs labeled onto the nanoparticles at the start of the experiment. Again, I'm just showing you a single slice here, but this is a three-dimensional assembly of cells containing probably around about 500 individual cells. And then what we do is we track the progress of what happens to the spheroid, this tumor, over time. And what we see, for example, 24 hours later, is that the drugs or the nanoparticles have started to pass through the individual cells and are now starting to appear in the central cells. And it's this particular set of parameters that were really interesting. And our technology is allowing us now to quantify and to ask questions about how this process actually occurs. So again, how do we do this? Well, of course, what we're able to do with our imaging technology is measure where the nanoparticles are within each of the cells within the tumors. And that's shown down here on the bottom series of images. Again, at the start of the experiment, we see that the nanoparticles carrying the drug are all at the very edge or periphery of the cell. After 3 hours, we see that those drugs have entered the outer cells. After 8 hours, they've got a little bit further, and by 24 hours, those drugs have started to penetrate to the very center of the tumor. And again, on a cell-by-cell basis, we're now able to measure this and literally count where the nanoparticles are carrying their drug inside these tumors. And now, of course, what we can do is we can start to see what are the mechanisms by which these nanoparticles deliver their drugs into cells. And we do that now by systematically examining the genes that we think may be playing a role. So here, for example, at the left-hand image, you see a control spheroid, a controlled tumor. Again, the nanoparticles are in red, and you can see the cell nuclei in blue. And you see in this experiment, which is 24 hours after addition of the drug, you can see the drug has penetrated into most of the cells within the tumor. However, when we stop certain genes working, as for example shown here, you can see that the drugs are either not able to bind to the tumor or the drugs very, very poorly penetrate into the tumor. And so what this work is helping us to do is to identify the genes, the proteins, and therefore the pathways that these nanoparticles use to deliver the drugs into the tumors. And of course, remember, we can zoom in on each individual cell. That's what we're doing in these left-hand panels, and examine now not at the whole tumor level, but actually at the cell-by-cell basis to understand what is happening and what is going on. And again, because this is imaging using very highly quantitative methods, we can now start to rank the importance of different genes with respect to this process. What, of course, this is ultimately allowing us to do is now start to answer this question of how these pathways actually work and how nanoparticles carrying drugs are able to travel between individual cells within the tumor and exert their effects. So this work is ongoing, and we're very excited by it because it's providing really the first important insight into how drugs are delivered into cells in tumors. But of course, what we also want to do is see what happens in terms of when that drug is delivered into those cells. So we're interested in looking at the toxic effects that some of these drugs might have when they're delivered. And so in order to do those experiments, again, we've been making a series of different nanoparticles that act as the carriers for these drugs. Don't worry so much about the detail of the slide. This is just to briefly explain. This is what these nanoparticles look like. You can see here, they're quite spherical in size. And what we do is we characterize their size, but also we characterize things like their charge and various other features. And in this case here, we're comparing two different kinds of carrier, one that's negatively charged and one that is positively charged. And we measure their sizes. We then add a fluorescent molecule so we can see them inside the cells. Again, now we add those nanoparticles to these spheroids, to these tumors. These nanoparticles now in this example are shown in green, and we image these tumors in 3 dimensions. So here you can see the 3-dimensional view of this little tumor. And then in this example here, what we're doing is we're adding a dye, this red dye here called propidium iodide, that actually serves as a marker for when cells start to die. So all of the little red dots that you can see in this image on the right-hand side indicates a cell that is dying, hopefully as a consequence of the delivery of the drug to this particular cell. And of course, remember, the power of our technology is that we can now zoom in on individual cells and start to understand the process by which cells are dying. So in this example here, we're looking at the lysosomes, the low pH compartments of cells shown in green. And what we can see is that those cells are dying. Cells that are highlighted in red all have very large lysosomes, again, telling us something important about what is happening to those compartments inside cells. In this case here, showing that the nanoparticles delivering their drug are inducing a swelling of those lysosomal compartments. And then finally, in the bottom here, we're again now using a different dye. In this case, this is a red dye that tells us something about the mitochondria, the energy-producing organelles inside these cells. And again, highlighting to us that cells that are dying have very, very poor ability to generate energy. Again, this is providing important information in terms of how the cells are dying. So finally, what we're continuing to do? Well, we're continuing to develop these methods that I've talked about. We're continuing to develop these spheroid models as models for tumors, as models for cancer. And actually, one of the biggest problems that we have right now is not really a problem of biology, it's actually a problem of data. I'm just going to finish by sharing some numbers with you. So for example, these images that you see here are small 4-day-old tumors, 4-day-old spheroids that we grow in the lab. And remember, we said we grow 700 or 800 of these per well of a plate, meaning that on average, we are growing around about 21,000 individual cells, which then equates per well, which then equates to about 75,000 spheroids, or 2 million cells in total. And the image data alone hit for this one experiment is 4 terabytes. And in fact, if we scale these experiments up and grow much larger tumors, much larger spheroids, the data sizes become massive, 12 terabytes per experiment. So you can see that right now, the problem we're facing is not really one of biology, it's one of data and handling huge data sets. So let's summarize what we've talked about in the webinar today. Well, we started by talking about an automated image and analysis platform. And we said that by studying cells and doing cell-based assays, this provides us a really powerful tool for understanding how cells work and understanding the pathways that are operating within individual cells. We talked about how some of the team members in my lab have been using high-resolution imaging to understand the biology of certain diseases. And then we talked a little bit about nanoparticles and how we're using nanoparticles to deliver drugs into cells, and how we've developed methods that allow us to study the penetration and movement of nanoparticles across cellular layers. And finally, we've finished up by talking about how we're now starting to study the toxicity of these drugs and nanoparticles, and trying to understand how individual cells are dying when we deliver these drugs to them. So I'm just going to finish by saying, why do we do this? Well, hopefully I've convinced you that the approaches we're taking are very, very powerful. And these approaches are really powerful in the context of the biopharmaceutical sector. And one of the reasons why we do this science and research in Ireland is Ireland is a very high-tech country. In fact, in Ireland, we have 9 out of the top 10 pharmaceutical companies and 14 out of the top 15 medical device companies. And they're very interested in utilizing this technology in going forward and in terms of developing new drugs. Where do we do this science? Well, we do this science here in the O'Brien Centre for Science, the College of Science at University College Dublin, a 30,000 square metre building, state-of-the-art, purpose-built for this type of science that I've described today. And our teaching methods and our research uses really innovative methods. So Ireland has really positioned itself as being a European leader in the biopharma space. And all of these companies that you see listed here are important employers in the Republic of Ireland, currently employing more than 30,000 people in the sector with an investment of more than €4 billion. And we just hope that some of the work that we're doing in my lab and certain other labs are contributing to these efforts. Ultimately, this is for me all about impact. The science, the research that we do is about providing innovative solutions in health and disease. And this is effectively just about my last slide, but it's talking about one of our alumni from UCD. Her name is Professor Teresa Lamb, and a number of years ago, she did her bachelor's and her PhD at UCD. And Theresa Lamb, Professor Lamb, went on to a very illustrious career. She now works at the University of Oxford, and she is the scientist, the lead scientist, who was responsible for the very first anti-COVID vaccine that was developed by Oxford University in collaboration with AstraZeneca. And her scientific career started at UCD, doing experiments in a similar vein to those that I've described to you today. So what do I see? I see really amazing opportunities for our science graduates right now. Today, of course, they're focused on the pharma sector, biotechnology, and how they can impact health and disease. But of course, we think Ireland offers really amazing opportunities for science graduates in many, many different places. And so finally, it just remains for me to thank all of my research team over the 15 years I've been in UCD for their amazing support and their amazing science that they're doing that's hopefully driving ideas forward in terms of what next generation pharmaceuticals might look like, and hopefully that we'll be able to solve important questions in health and disease in the very near future. So with that, I will conclude the presentation in the webinar.
Thanks, Professor Simpson. So we do have one question from Hannah from Indonesia. So the question is, does this confocal microscope use specific dye to stain the cell, such as the flow cytometry, maybe?
That's a very good question. Yes. In my lab, we mostly use microscopy rather than flow cytometry. And one of the main reasons for that is that we're interested not only, for example, in what's on the outside of the cell or what markers might be there, but we're also interested in what's going on the inside of the cell. And we're interested in looking how different structures inside the cell change, for example, when we add a nanoparticle carrying the drug. And so for that reason, it's important that we use microscopy so we can visualize all of that process. And so what that actually means is that we use a whole range of different dyes and fluorescent reagents that allow us effectively to look at lots of different compartments at the same time. So for example, on one day, we may be interested in the mitochondria to look at the energy metabolism of cells. But equally on another day, we may be interested in organelles such as endosomes or lysosomes to study the process by which drugs are being taken into cells. So I think really, so the short answer to your question really is that we use a range of different dyes and tools to visualize different events going on inside cells.
Thanks, Prof. Simpson. Hope that answers, Hannah. Also, like, out of my curiosity, like, because I'm not from the scientific background, because you mentioned before, like, the nanoparticles is kind of the vehicle to make the drug smart, right? Does it mean like the nanoparticles can increase the effectiveness of the drugs or medicine itself?
Yeah, that's a very, very good question. That's exactly what we're hoping. So one of the big problems that the health sector faces at the moment is that we have great drugs, great pharmaceuticals, but often they're very toxic. So for example, if you have chemotherapy, you probably know there's often many side effects. And those side effects occur because those drugs, those pharmaceuticals are very dangerous, they're very toxic. And yes, they can kill the cancer cells, but they can also kill the normal cells. So our hope is that nanotechnology provides the possibility to take those same drugs, but to encapsulate them in a nano device. And on that nano device, we would put information to tell that particle only bind to a cancer cell, don't bind to a normal cell. So that would provide a mechanism by which nanotechnology could deliver these anti-cancer pharmaceuticals only to the cells that we want to kill. So therefore, that would really increase the targeting, and that would therefore increase the precision of how these drugs are actually being used.
Interesting. So does it mean like it can be like shorter the duration of the taking medicine? Like for example, for TBC, we have to take medicine every day, like for months and for years, right? Does it mean like can be shorter with the nanotechnology?
I think it could affect it in two main ways. So one is certainly the length of time of treatment you might need. And I would say the other is the amount that you would take. Again, one of the problems at the moment is that we have to take very large amounts of many drugs, simply because that drug is diluted through the body much of that drug is going to go into the wrong cells. So again, our hope is we could use much, much less, maybe 10 times, maybe 100 times less than we're using now, because we know that drug will hopefully only be targeted to the cells that we want to die. And again, that would then have really positive effects in terms of reducing the number of side effects from these drugs.
Good one. Thanks, Professor. So, do you have any recommendation if student say that student from Indonesia that plan to like learn the nanotechnologies more deep into that?
Yeah, so what I would say is that some of the things I've talked about in the area of nanotechnology today actually involve not just biologists. So I wanted to give a hint of that, yes, we are a biology lab and that is our focus. But actually every day, as maybe you saw, we work with colleagues from chemistry background. We work with colleagues from computational backgrounds. We work with colleagues from data science backgrounds. And actually that's really important because if we're going to develop this technology and make it successful, and if students are interested in this technology, you can come from many different backgrounds. So again, the programs that we offer in UCD in the postgraduate space, for example, we have programs in biotherapeutics, biotherapeutic design. We have programs in biotechnology. We have programs in biomolecular science. And all of these programs offer something slightly different, and they all would take in students from slightly different backgrounds. So effectively, we're just looking for students who've got a real passion for this area, who think they can go, maybe make a difference in the world in terms of health and disease, can come and learn some advanced skills in this wide area of nanotechnology and therapeutics, and then maybe contribute to the health sector, as I talked a little bit about at the end. So I think really amazing opportunities right now.
Good. And how's the job opportunities like? Is it can be applicable to only to healthcare industries, or can be applicable to other industries as well?
Yeah, exactly. So at the moment, this is the economic situation in Ireland is extremely strong, and of course, all students who do a postgraduate degree with us get a 2-year study visa at the end. Actually, right now in Ireland, we have almost no unemployment. So actually, all of the students who are graduating, both our Irish students and our international students, actually, ultimately, they end up getting a job and working. And what we see is that, yes, many go into the health and pharma sector, but actually many go into businesses. A number of students go into IT, for example. That's a very strong sector within Ireland. So I'd say again, at the moment, there's really, really good opportunities in Ireland right now. We are the gateway to Europe and the European Union. So do think about the opportunities that Ireland offers.
Okay. Thanks for the insights, Professor Simpson. So I think there's no more questions. Any other comments or maybe tips for students that plan to study in this field?
Well, I think my I think my main tip is it's really important for students to follow their passion. I think there's lots of opportunities in what I've talked about in the broader biopharma and life sciences sector. We're generally looking for students who are interested and passionate for the subject. And if you come with passion for the subject, then you're highly likely to be successful in your career. And again, given everything that I've talked about today, some of the examples I've given, and obviously the wider information about the opportunities in Ireland, I would say to students, do your research, talk to colleagues at AECC, and just have a look at whether the possibilities that we offer might be best for you. And again, if anybody's still not sure, I'm sure colleagues at AECC will provide additional information. Or of course, students are also very welcome to reach out to myself and my colleagues in the College of Science at UCD, and we'll be more than happy to answer any further questions.
Sure, great. Thanks, Professor Simpson. So let's call it for today. Stay safe and safe flight for your flight to coming back to Dublin. Right?
Thank you.
Bye. Happy weekend.
Bye. Thank you very much. Bye, everybody.
Take care. Bye-bye.
the easiest way to study abroad. Guys, you don't need to worry about finding information on studying abroad. AECC will provide all the information you need. AECC has been helping students from all around the world to realize their dreams of studying abroad and has industry partnerships and local know-how to help you get set up with things like banking, insurance, accommodation, and more.